Now, you can see today we are dealing with chemiosmotic hypothesis. Now, this hypothesis actually explains the synthesis of ATP, but it is linked to the development of proton gradient across the membrane. Now, you see, this hypothesis is basically common to both respiration and photosynthesis, but there is a little difference to it. Okay. Now, if we consider the site of chemiosmotic hypothesis, or this phenomena, it takes place in the thylakoid membrane. What is thylakoid? You know, stacks of thylakoid are found in the grana of the chloroplast. Okay, so this thylakoid membrane is actually the site inside the membrane. Okay, but in case of respiration, the site is the intermembrane space of the mitochondria. Okay, that means the intermembrane space of the mitochondria is responsible for chemiosmotic hypothesis or the production of ATPs. Okay, against the proton gradient. See, here we are dealing with proton. Okay, and what is gradient? Gradient is actually the concentration. Now you see. Now, see, the process starts with the activation of electron. See, the electron has to be activated and it has to be transported through electron transport chain. So, electron transport chain forms the primary and the functional center for chemoosmosis. Now, what happens? It takes place in three important steps. What are the three important steps is, first of all, you see, within the thylakoid membrane, you know, this H2O, water, is split into hydrogen and oxygen. Okay. Now here you see the electron is released from one membrane and is passed on. Okay. So this release of electron goes into the second step. Okay. So inside the lumen, so once it is electronically charged, okay, the hydrogen and the oxygen is there, this electron is accepted not by an electron acceptor, but here the electron is accepted by a proton carrier. That means what? This disturbance, you know, this hydrogen has to go out, but it is not, the acceptor is not an electron acceptor, but a proton carrier. So in this case, the hydrogen has to be removed. Okay. The hydrogen has to be removed, and here hydrogen is called as a proton. Now here you see, when the electron is released from this oxygen, this electron this happens in PS2 first, isn't it? From PS1 to PS2 it goes. Okay. So first reaction takes place in PS2. So this happens in PS2 and this electron is finally accepted by this. So when it is released, see, when it is released, see, the proton is finally reduced to NADPH. So this disturbance, you know, from PS2, this electron is released and this PS2, photocenter 2. Okay, in photocenter 2, when this electron is released and goes to photocenter 1, this finally, you know, there is an enzyme that is known as Atp reductase. So this Atp reductase finally, you know, it reduces this NADP to NADPH. Okay, NADP to NADPH. Clear? So it actually forms NADPH positive plus H. Okay, this actually hydrogen is released here. Okay. Now you see, here the third part is, you know, the Atp synthetase is also another enzyme. You can see this is the stroma and this is the grana. Now you see, in stroma, the proton is reduced. Why? Because since the water is split and it moves to the membrane here, inside the lumen of these thylakoid membranes, so concentration is reduced here and it is increased over here. So this decrease and increase causes a concentration gradient. Okay, so breakdown. Now pump or a proton pump in which they convert this energy, this proton outside the membrane. So see, normal condition the hydrogen is going here. The concentration of the proton is inside the lumen, but this pump is facilitated, so that breaks down and brings down the proton outside the membrane where it is finally joins, okay, to with water, okay, oxygen. So this thing, since it is going against the proton gradient, it synthesizes ATP that is produced here in the photocenter too. So this breakdown of gradient leads to the synthesis of ATP. This side it is synthesized once the breakdown of ATP gradient is there. So against the gradient, so ATP is used for the proton pump as well as the ATP is also produced here. Here it is used up in the proton pump. Okay. Now if you see in your book, if you can see this diagram, you see there are two parts, PS1 and PS2. Okay, so initially the PS2, since it has got, it activates later. It is 680 and here it is 700. Okay, so here they have got a cytochrome complex. Okay, and you see the ATP pump here. The potential gradient within the lumen is high and outside the membrane, outside the grana or the thylakoid membrane, that means within the stroma, the concentration gradient is more. So to pump the hydrogen out from here, they need a potential gradient. So here the ATP is used. Okay, and within and to break down the gradient, ATP synthesized is also there. So this is actually known as chemiosmotic hypothesis. Okay, so here again this proton gradient is important, since the proton gradient is done across the gradient breakdown, it synthesizes ATP. Okay.